Hey there, everybody. Welcome to the webinar. We're about to get started in a few minutes, but before we do that, let us know where you're coming in from. This is a good cross ocean webinar. We got New Zealand on one hand. We got here in California on the other hand. For those of you joining us in New Zealand, kia ora. It's probably a pretty nice day over there. It's kind of this. What is it, like late spring over there? Or is it early summer?
Yeah, it's kind of the season of mud right now, so
The season
rain.
of mud.
Yeah,
Nice.
mad.
Eventually they'll just converge. Both here will be this. We'll be on the season of mud on both continents.
Yeah,
Well,
it kind
awesome.
of felt that. Yeah, same temperature. I think when I left Montana is what it is here now.
Oh, nice. Okay, so for those of you who are new, if you have questions, you can type them. There's a little chat box below. Unfortunately, you can't see other people's questions, but we'll be able to see your questions. And we'll start out by tell us where you're coming in from. That would be awesome to know. Trying to see. I see a few familiar faces here, like Svetcha and Siska and Andrew Langford. But I know there's definitely a few people where this is their first webinar or first time they've ever done a webinar. So we got Ellen coming in from Australia. Welcome, Ellen. I'm waiting to hear from other folks. Nicole, where are you in New Zealand right now? Auckland?
Yeah, right now I'm in Auckland. My family's
Nice.
here, so catching up with family.
Great. Cool. Personally, I'm coming from Berkeley right now, and just staring out the window, I was like, I want to be outside. It's so nice. It's so nice. Trapped inside. Okay, everybody. Well, we might as well just get started with the webinar. Without further ado, let's get on with it, So, everybody, welcome back to Sustainable Design Masterclass. We're dedicated to bringing you some of the top regenerative system designers across the world. We've had a lot of great system designers from Australia and New Zealand, because we know that's where permaculture pretty much originated, with David Holmgren and Bill Mollison. And there's a lot of people that have been working there for decades. And it was great getting approached by Nicole, because she's working in the world of repairing soil biology. You don't hear a lot of people who are doing that besides, Elaine Ingham and Gabe Brown. And so it's exciting to hear her perspective on restoring soil biology. Now, if this is your first webinar, you want to turn off your distractions. This phone you have right here, it's not your friend. It's not your friend for this webinar, it's your enemy. So just here, boom, toss it in the garbage. Just delete your Instagram, at least temporarily. Turn off your farm Tinder, whatever that is, if you're using that, because you want to be present. You want to just soak in all this amazing, just wealth of knowledge, because this is going to be great. So without further ado, we'll get started with the presentation. I'm excited to introduce Nicole Masters. She has a company called Integrity Soils that's based out of New Zealand. To date, they have helped biologically convert over 1 million acres of land now into biological agriculture without chemicals, without pesticides. It's pretty exciting. And she definitely takes a soil food web approach to everything, converting it from chemically based agriculture, ranches, pasture, to organic, using biology. And she's been doing this work for about 20 years, and she's been consulting worldwide. So you're going to hear a great perspective on restoring soil biology on a whole range of scales. So I'll turn it over to you, Nicole, and thanks for joining us.
Oh, thanks for having me, Rolly. I really appreciate being here. Yeah, so welcome everybody. I think I'll just get started a little bit with some of my background, and I'll kind of do the screen share. Can you see that, Rolly?
I can't see your screen at the moment, but it should just pop up right now. Okay, cool. Now it's there. Awesome.
All right. So my background is actually ecology. I majored in soil science, and when you study soil science at university, there's a lot of chemistry and bulk density and cation exchange capacities, and it wasn't really until I was actively doing vermiculture and getting into the worms that I discovered soil biology. And I was lucky enough to go to a workshop with Elaine Ingham, and she mentioned microbes, and I just I've just done 3.5 years of soils, and no one mentioned microbes the whole time. And and if you look at the soil science books that we're being taught at universities, New Zealand's manual is, a couple inches thick. There's 4 pages on microbiology, and it was like a light going on. And I think, many people have that kind of experience, and I think a lot of people have a story, what's led them to soil. And for me, my story started when I was poisoned with paraquat when I was 15. And, I was hospitalized and had just ongoing problems because paraquat will pass the blood-brain barrier. And so what was interesting is I didn't know at the time that it was paraquat, and it was about 15 years later that came to be revealed. And it's been so interesting to me that I've spent my life working with people in terms of how do we reduce the chemical loading that's in the environment and in food without actually knowing that I had a chemical loading myself. And I guess I knew that intuitively, so in the back of my mind that was going on. So so this image here is of some of the operations that I'm currently working in. So soil is what links them, but pretty much you can think of every ecosystem, ecotype, climate, sector, they all have soil, and soil is what's connecting them. But it means that I feel like I'm a magpie, like I'm always kind of collecting shiny things and collecting ideas and looking for, well, what's working here in a dairy farm system in New Zealand? How might that apply to a market garden in California, for instance? Or, how does this work in pip fruit production? Or what are we looking at when we're looking at golf courses, for instance? So I have a very diverse client base, and it is certainly incredibly interesting working with different outfits. So I'd like for this to be a conversation, Rolly, if we can, have a bit of back and forth, because there's so much that we can cover. So my particular approach really is looking at regenerative agriculture. How do we regenerate a resource? And I guess before we get into that, I'd just like to define it for a minute. So regenerative agriculture, if you look into the literature, it's defined as any system of food or fiber which incorporates natural processes into ag production, ensuring that, one, we're profitable and that we have efficient systems and we're producing quality food. And I think that's where we really need to pull ourselves back, is it's an outcome based, as opposed to a system that might involve certification of we're treating animals humanely, or we have no residues, or whatever it might be, or we're not using chemicals. It really looks at what is the outcome of your production, and is it actually returning a profit to producers. It's also looking at how do we reduce external or harmful and non-renewable inputs. Now, I guess you got to look at here, it's about reduction. So there's no places that it's a no. Like, I got in an interesting conversation yesterday with a soil scientist who was adamant that phosphate is always going to be a declining resource because we're exporting, all that blah, blah, blah kind of conversation, and, ultimately everything ends up in the sea. Nobody's saying that we're never going to use something. So at some point, you may need to intervene, like it might be with antibiotics, or you may need to intervene because you're about to lose a crop. So you get to the point where you actually have to make a decision. Maybe I need to use a fungicide here, although I'd like to think we don't need to do that. But there's no no's in this. It's how do we do that in a way that we're producing the best quality food? How do we look at how to reduce these external inputs? Because we're looking at, if we can improve the health of an ecosystem, then we don't require fungicides and pesticides. We don't require as much herbicide. We're not, and we're starting to support the whole, food production. So the aim is, how do we reduce costs at the end of the day? Because if you look at what's happening in farming and agriculture around the planet, producers are no more profitable. With all of these great big leaps forward that we're having in technology, it's not necessarily rewarding the farmers, it's rewarding the supply companies and banks and everyone else that's around the farmers, but they're not necessarily any more profitable. And that's part of regenerative agriculture, is addressing that. Now, the third point to regenerative agriculture is it's about participation of farmers in the rural community, that it's not top down. This is very much bottom up. And you look at the leaders involved in regenerative agriculture, it's very much coming from the ground up. And we've been fortunate enough to work with some really brilliant scientists and extension agents that are willing to be part of the conversation, engage with producers, as opposed to telling producers what to do. So I really like this quote from Professor Norman Uphoff, who says that postmodern agriculture is not anti-science. When we talk about postmodern agriculture, we're talking about regenerative and biological and your permaculture, because it's the most modern agriculture, because we're building carefully and creatively on advances in scientific knowledge, particularly in the disciplines of biology, ecology, and microbiology. So it brings in these aspects of biology. And some people say, well, are you farming like we were 120 years ago? We're going back to what our grandparents are doing. And for me, we're not, because we are developing a deeper understanding of biological interactions and mineral interactions and their synergies, how to do things in a more measured way than just, say, putting manure on a crop. Yeah, so it's not taking a step back in time, we're not throwing everything out, basically, is my point with all of that. So the root of regenerative agriculture is really asking questions of how does nature work and why, and asking those why questions on every part of our operation. Why do we need this additive? Why are you using a pesticide? And really getting to what is driving that, and what drives all of it, and what's driving nutrition and plant defense is these communications that's happening between the plant and the root and the microbes. So how do we build what we call these are our Rastafarian roots? How do we build root systems like that, or a rhizo sheath like that, that's starting to defend and protect plants against stress, and starting to enable us to build resilience in a changeable climate, so that, say, we do go through a dry period, or you have a waterlogging event, or there's some kind of stress to that system, that there's momentum that plant can then continue and recover quickly, which is all about this communication that I'm going to get into. So when we look at soil health, we don't just consider one aspect of it. So we came from the last 100 years of just really focusing on chemistry. And before that, I mean, they were looking at humus and organic matter, but just focusing on one element. It's all about how do these different things interrelate? What is the impact of, say, having a high clay soil, so your soil texture, or what kind of chemical imbalances might you have? So for me, I pull on everything. Like, I mean, I've studied permaculture and biodynamics and conventional agriculture, and pulling on what for me works well. So I'm not saying I've got all the answers, and I don't think that anybody does, but we're all learning this as we go. So for me, microbiology is not always the answer, or minerals aren't always the answer. It's looking for what is it that's putting a drag on this particular system. How do they all interact to form soil health? So I look at what I call the 5 M's. So it's your management, your minerals, your microbes, your mindset, and your organic matter. So what is it that's really putting a drag on this system right now? And then look at how do they interact, and what is actually slowing you in achieving your results. So I work with some of the leading lights in holistic management, who have been doing some incredible work with grazing for the last 30 years and then kind of plateaued. And it's like, well, why did you get those early gains in production and now things have just kind of fallen away, or just, they've just flattened off? And it's looking at, well, there's more than just management that's involved in this. What is happening with some of your microbial interactions? What is happening with minerals? And starting to look at what I call tickling the system. Where do we find those catalysts? Where do we find those points for leverage so that we can start to achieve our goals in soil health? So I'd like you to consider, and you probably do anyway, and particularly if you've got a nutritional background, you'll get this, but soils are very much like what we're dealing with in the human gut system. So, you bite an apple and you chew that and you make it smaller. Well, there's arthropods and microarthropods and invertebrates and things that are in the soil that make that apple and break it down smaller. Then it passes through your gastrointestinal system. In there are enzymes and acids that work away to start to break down that material. So in the soil, we have very, very powerful acids and exudates and enzymes that are all doing this process to break that food down smaller and smaller and smaller. And while they're doing that, they're producing things like vitamins and enzymes. And these are things that are catalysts for health. So what we're doing effectively in this gut system is it's the microbiology that are getting hold of the food and are then making it small enough to pass through to the bloodstream. So this same process is happening, but in the plant, I want you to consider that its gastrointestinal system is outside of its body, that the plant, instead of having all of its systems all in one, it's basically farming out these systems. So it's saying to a specific microbe, I need water, for instance, and sending a signal, or I need this trace element, or right now I'm under attack. And there's some really cool research that's been coming out right now that's looking at induced systemic response, for instance. So a plant might be coming under attack, it sends a signal to biology, and the biology respond and produce different types of metabolites that induce a defense inside that plant system. And I think, Rory, you've had other people talk about this, but consider that a plant's digestive system is outside of its body, right? So we've got all of this microbiology, huge numbers of microbiology, of which we can't even, I don't think we're even beginning to scrape on how many they are. I mean, the latest counts, there was 37 trillion different types of organisms on the planet, and we've mapped about maybe 1 million. Like, our understanding, I heard a great quote recently, it's like we're standing on the edge of the void looking out into the darkness, and we're shining a torch or a flashlight in American cases out into that void, and what we can shine on is what we're focusing on, and that's all based on our current scientific understandings or the questions that we're asking, and we're missing most of what's out there because we don't understand it and we don't know how to measure it. So this is part of why I find soil so very exciting, is we're on the cutting edge of a new frontier of understanding. So we've blown this microbial bridge, right? So it's these microbes that are helping us grow, they're regulating health, providing vitamins and enzymes, and basically enabling you to be healthy, okay? So we've broken that down in the human health through lots of different mechanisms, and I'm sure people have probably spoken about this with you, but in terms of things like antibiotics, or not being born through a birth canal, or not being exposed to soil early on, or not having cats and dogs, or eating preserved foods, all is having an impact on the types of microbiology in gut systems. So you see people have a lot of candida, or they're addicted to sugar. That's changing the microbes that are in our gut systems. So they're now relating the human gut microbiome with pretty much everything. So over 80% of disorders now are being related to a disruption in the human gut microbiome. And what's cool is the research that's happening in the human gut is then enabling us to understand deeper what's happening in terms of soil microbiology, because a lot of these processes are similar. Well, I mean, your soil's not going to get acne, but there's other things that's going to happen in terms of soil health. So I want you to think about, like, when you're in when I was in the US, I was really surprised to see this, that you walk down an aisle and half of the aisle are dedicated to things to make you poop, and half the aisle is dedicated to things to stop you pooping, right? The American gut system in particular is totally disrupted, all right, from these different things that we're doing. And we've done that. How
dare you make fun of our pooping problems?
Oh, I know, you guys and your pooping problems.
But it's true, there's a lot of pooping problems out there.
There is a lot of pooping problems. I mean, yeah, I think at least 30% of the American population have major pooping problems, 30%. Yeah, and I guess if you look at glyphosate too, the amount of glyphosate that's in our water and in our food, it's an antimicrobial, so it's disrupting the gut system. So there's lots of ways that we've disrupted it, and I'm sure none of your listeners would be experiencing this. But yeah, so we're doing the same thing to our soil. So our soils basically have indigestion, constipation, gas, and diarrhea. So if we look at the New Zealand environment, a lot of our soils here have rampant diarrhea, and you can see it because it's coming out into the waterways, so just excreting huge volumes of nutrient. Whereas if you go up into the Midwest, we've got soils that what I call are just constipated, all right? So they're just holding tightly onto nutrients and they're not going to give it up. So what I want to show you here is an example of two soils that are side by side, where one, the gut system is really disrupted. So this particular ranch is in Nevada in the high desert. What we're looking at, if you look along that stream, that riparian strip splits these two fields that I'm going to show you. So the field to the south, which is further away, has had 120 years of being cut for hay. It's got an irrigation system that's broken. So at the bottom of that, not the really far hill, but the first hill you see, there's like sagebrush. There's an irrigation system that runs along the bottom of that. It's 120 years old as well, and it's broken. So in some places they're getting too much water, and other places they're not getting enough water. Whereas in the foreground, what we're looking at is fields that have been holistically grazed or adaptively managed, however you want to call it, for the last 30 years. So they took the sagebrush out, they have optimal irrigation, and they're being able to water it perfectly. So the management on this north side, so the bottom, is being managed a lot better. So they've been doing working with holistic management to repair the riparian zone. So this is what areas look like in 1973 compared to now, and you can see that all across the watershed that they've done a fantastic job in repairing riparian zones. So it's not excluding animals, it's just allowing animals to come in at different times of the year, heavier animal impact, and then ensuring that they don't camp in these areas. So the beavers have come back, the water table's coming back, they're seeing water tables lift, and so things like tall sage, which was in that photo in 1973, was all around there, is now dying. So you just see these kind of husks of this tall sage as the water table comes up. So these guys know how to manage. But what's happened, so this is for the last 120 years, so we're comparing the south side to the north side. So 120 years of degrading irrigation, hay cutting. At times, so this photograph, they have 270 horses were being grazed on these lands. So a lot of people think that horses are really destructive. It's not the horses per se, it's how we manage them. All right, so this is what the fields to the south look like. We've started calling them the Bronx, because it's the south side. It's where you don't want to be going. And what you see, all of that green vegetation there is it's a reed known as poverty grass or wiregrass. So it's a it's a non-mycorrhizal plant species. So what was interesting, and if you can see kind of where they've dug the trench, that soil just comes off like a carpet, like it's just a mat of root systems on top, and then if we look deeper, it's bone dry. So when we dug this hole, the rancher was just shocked. So the soil is dust, yet 2 feet down you've got a running river of water. So there's a huge amount of water there, but the soils are totally what we call hydrophobic. So if you look at that right-hand side picture, what we've done there is done our water infiltration test. So basically you just bang in a piece of downpipe will work, or a metal steel ring. You can look online to see how to do these infiltration tests, really simple. We put in 1 inch of water, and after 24 minutes we kind of you could see it wasn't going in anywhere, so it went in probably that 1/5 of an inch, and then it just sat there, and it's just bone dry. So these soils are what we call hydrophobic, so they're literally afraid of water. They're not going anywhere quickly. So this is what we're seeing all around the world right now, is we have soils that are water repellent or literally afraid of water. And so what we get is this drought flash flood situation that's happening all over the world right now. And so it's not because of climatic pressures, although that's not helping, it's that our soil systems are broken down. And so you can easily do this test for yourself. You can even do it in your garden and have a look. You want to see that water go in. 1 inch of water should go in faster than 12 minutes minimum. So at 12 minutes, your soils are broken. And that's for the first inch. It's the second inch that counts. So we didn't even bother with the second inch on these, and we have done other tests like this there, and the water just doesn't go in. So 80% of what was growing is what we call these non-mycorrhizal species. So your non-mycorrhizal might be things like your lupins. They're non-mycorrhizal species that will live in the water, so reeds and rushes, and then your chenopodium, and so like your beets, your spinach, lamb's quarters, fat hen, or your amaranthus family, and then your brassicas, right? So canola or anyway, you know what brassicas are. So cauliflower, broccoli, all right? They don't have this mycorrhizal relationship, and I know you guys have spoken about mycorrhizae quite a few times, right? So then when we dug a hole on the other side, so this is what it looks like above ground. That's the rancher in me, but I just put it in because it was the best photo I've had. It's Garrison creeping foxtail, so it's or creeping Garrison foxtail. It's a plant that does like a lot of water, but you see this is not, I'm not saying that this soil's perfect, but yeah, it's got really good ground cover. This is what the soils look like when we dig holes, right? So see the difference just in color. So you've discussed before, but the color, a big part of that color is humus and biological metabolites. So how we're building that water holding capacity, how we're building nutrient holding capacity. So it took 1 minute for 2 inches of water to go in. So that first inch went in about 30 seconds and the second inch went in 1.5 minutes. All right, so they've restored their water cycle. And there's less than 5% of those unpalatable species as such. So species that, you know, the cows aren't really going to get into. But you can see that depth of profile and see what we call these Rastafarian roots starting to build. One thing that we looked at, so when I'm looking at an operation, I'm going to look at what's happening with your soil minerals as well as your microbes, and then look at plant tissue tests. So the thing to look at here was in the south side, the calcium dropped, and we measured above the irrigation line so we could have a look at what was happening there. And basically 2/3 of that calcium's been washed out, and it's been lost due to poor management and lost due to compaction and cutting for hay and overgrazing. They've lost their magnesium, lost a huge amount of potassium, and actually the potassium on the north side is in excess, and very low in sodium. And sodium is an interesting one because what you'll find in a lot of old landscapes like the US is sodium's often moved from higher areas and it's been leached down. So then people get all worried about sodium when it's lower down because, you know we've got a sodic soil or a saline soil. But actually most of your soils in the US are sodium deprived and sodium deficient. And you'll see that because animals are looking for it. And it is actually an essential element for animal performance and microbes. So the bottom bit there is just looking at the difference in pH and then the cation exchange capacity, so the size of their bank account. So when you lose your humus, you actually lose the size of your bank account in terms of how much nutrient can be held. So I'm not sure if you guys have been looking at any kind of chemistry, but I'm just putting that out there just to say that they've leached and lost their bank account. How much nutrients is sitting in that soil has been lost through 120 years of really, really poor management. So we did the biological tests. There's a lot of numbers there. I don't know if I necessarily need to get into the numbers, but basically the north side had 10 times the amount of active fungi and it was still low, right? And part of that is because those soils do get so wet because they are irrigated and the irrigation isn't perfect. But the total fungi were 3 times the amount, and that total fungal number is perfect for growing good quality grass. Active bacteria, about 1.5 times. They had about the same amount of total bacteria, but the total fungi to total bacteria ratio, this is the one that's really critical, and actually the one on the left should be in the red. This is the one that you would have been looking at with Dr. Elaine Ingham in terms of what are we setting the scene for? Are we setting the scene for, in this case on the left, non-mycorrhizal, weedy, low-quality species, or are we setting the scene to grow good quality grass in this situation? So if this was for an orchard and you're growing blueberries or whatever, then we're going to want to push to more fungal dominance. But so we did this testing just to look at what is the status quo, what is the difference between these two soils. So that just sums up these differences that I've said. So that total fungal to total bacteria ratio on the left is 0.57, so perfect for your low successional weeds. On the right at 1.75, so perfect for pasture. So this is what we're seeing more research coming out about, is how do we lift this active fungi? How do we get more fungi in the system and start to build that aggregation? They're flagellates, so this is your protozoa, and I feel like I can just talk about this. You guys have done enough now, hopefully, to have this in your head. So the flagellates are absolutely critically low, and they're really important for nutrient cycling, really important for controlling your fungi to bacteria numbers. Yeah, and they're super low. Arbuscular mycorrhizal fungi at 0.09, that's not the lowest that I've been measuring. I've been measuring soils in North America that are down to 0, so they have no mycorrhizal colonization whatsoever. These guys haven't been putting on things like soluble phosphate that might actually really kill that mycorrhizae. They haven't been cutting the soils up. It's just really compaction. So mycorrhizae will survive quite well in very dry environments. It's still too low for what we're looking for. However, it wasn't as big a difference as I thought it might be. And there's a lot of root feeding nematodes on the south side. So what will often surprise people if they dig holes and start to look at root systems is often there's more pests eating roots under the ground than there are animals above the ground eating. So there'll be things chewing on roots, and nematodes would be one of those things. So that's something critical that we want to look at. We then did plant tissue tests. So here we're looking at what is happening in terms of quality. So the north side had over twice the amount of crude protein. Protein obviously is important for weight gains and milk production and things. An increase in calcium. So pretty much if you look at Bronx, everything's in the red. Everything's in the red in terms of phosphorus, magnesium, potassium, but pretty much every element, so trace elements as well. So not really a surprise, but it's just an example of what we're looking at is we're trying to identify what is it that's not coming through to the plant, what's putting a limit on even producing enough organic matter or having food for microbes and food for animals. So right now, if you wanted to just graze cows on that, and this is what we look at with adaptive management, is being able to have high impact. We're not growing any grass. Those soils are water repellent. They are very, very poor quality. So, you couldn't get animal impact on a soil like this to get started with without compromising animal health. And so I'm interested in how do we do this without having a negative impact on animals. So this is something that we looked at doing, is we put 30 pounds of vermicast, so worm castings, actually down the drill with some seed. So we're using that seed or those plants to interrupt the current cycle. So that photograph there is just those little seeds coming up. We've only just started doing this. But what's in vermicast is biology that will actually eat the hydrophobic waxy coating. So what happens when soils get very dry is they start, bacteria produce a waxy coating to protect themselves, but what it does is it makes that soil now water repellent. You could, in theory, harrow it or rip it to try and open up the soil, but actually that's a mechanical solution to what is a biological problem. So I'm always looking at how do we address what's the root cause of this. So it's biology that are causing this hydrophobic condition. So in worm castings are specific types of bacteria that will eat the waxy coating. So we're kickstarting that, and by opening up that soil, we're getting a root down there. We're starting to interrupt what's basically being shut down in the soil. We're also applying.
Nicole, real quick, did you mean 30? Is it 30 tons an acre? 30 pounds?
30 pounds. 30 pounds
okay.
Down the drill. So tiny amounts, and we're just mixing that with the seed. If we put stuff down with the seed, we can get away with a lot less than if we're trying to put it above ground. And all that I'm looking at is how do I influence that plant? How do I influence that plant root? Because it's the plant that builds soil, or it's the plant microbe interaction that builds soil. So if we can support that plant to get going, now it can get its rooting system down through that soil. Now we can have water entering that soil, which wasn't happening before. And now we get absorption. So we put on a little bit of fish and molasses and some seaweed. Seaweed's helpful for, it's got rooting hormones in it. Fish hydrolysate, feeds fungus like no one. It's just amazing. And we also know that, the bacteria weren't as active as they could be. So we're putting a little bit of sugars in for the bacteria. We also looked at balancing their sodium and their calcium. Most soils you can't afford to try and really address a lot of the mineral imbalances. And I know people are trying to do that, like force the numbers. All we want to do is start to create the environment so the plant can start accessing nutrients and start working with biology to do that. But if you don't have some of these trace elements and you've got really, really low sodium and really low calcium, which is the foundation for health, it's going to take you a long time to kind of get there. So I'm not saying you won't get there just with biology, but because they've actually effectively robbed Peter to pay Paul in the last 120 years, and they've been leaching out that environment, they just don't have the foundation right now. Now we can start to build that foundation with biology, but I'm not that patient. And with a soil that was in this porous shape, we put some lime on, so we actually put ag lime on at 2 tons, 2,000 pounds an acre. And then that's going to give you that foundation for health, because biology need calcium, the plants need calcium, everything needs calcium. And then the same with sodium. So that's where we've gone with that. So that will be a work in progress. But the take home from all of this is just how poor management can really, really degrade a soil environment till it gets to a point that even with the best management, that soil wasn't going anywhere. I mean, it had a lot of holistic grazing or holistic management people out there to have a look and try and work with grazing, and it wasn't really addressing, because grazing wasn't the limiting factor. Right, the limiting factor was biology was stopping the water even getting in, and the system was all leached out. So it's always looking at different operations in terms of what is your limiting factor. Okay, so so we go back to that microbes, minerals, management, mindset, and your organic matter. All right, so these guys are right up the top of Alberta, Canada, and I've been working with them for nearly 3 years. They are in the top 1% of performers in the region, and the reason that they came to me was that they had really tight compacted soils, and they could see that that issue wasn't getting any better, and actually it was getting worse, and it was costing them more in terms of fossil fuel use just to get across places. And so this, I'm interested in working with a lot of these bigger operations in terms of these are the ones, if we want to get chemicals out of the food system, these are the guys we need to be talking to. And then what comes out and the innovation that comes out can then help support some of the smaller producers in, say, third world countries who look to, you know, America as the example, or high performance agriculture as this is where they want to be. And it's like, actually, you don't want to be here. You don't want to be creating soils like this. So these guys, I really love working with them because they're willing to ask the questions, and they haven't been driven by crisis. They've been driven by, they can see where the future of agriculture is going, and they're not committed to that. They want to do better. So even though they've got organic matter in the soil, it wasn't functional. So there's a difference between having just organic matter sitting there and having high functional humified soils, you know, beautiful crumb structure. They had low biological activity, low mycorrhizal fungi, very, very high magnesium. And what's interesting about magnesium, it pulls a soil and makes it tight. So they're up to 40% magnesium in places where you want maybe 15% magnesium. So that becomes a limitation for biology, because if your biology can't breathe, it's really hard then for them to get down through that profile. And so this is where we'd be looking at minerals as part of that, in terms of how do we address that mineral imbalance so that biology can really get going. And trace elements are part of that, and these guys had really low sodium. So the hard pan was one of the first things we had to look at. So these guys had a hard pan that was as hard as concrete. Now, I had a video, but I don't think it's going to play. But yeah, that hard pan was like concrete once things dried out. They had major deficiencies in boron and manganese. And so those things are what we call our mineral catalysts. You only need a tiny amount, and it might be something we just do at the start just to kickstart a system. So for those of you that don't know about what boron does, for instance, boron is what takes different elements around the plant. But one of the things that it does is it opens the pump at night for the sugars to be pumped from the top of the plant out through the roots. So if you don't have adequate boron in the leaf, then that plant's not feeding biology. And so you could put biology out all that you want, but if that plant isn't able to actually start feeding the microbes, then nothing's going to happen. You're just going to stall. And so you will get a positive response from stimulating more microbiology, but it's actually not your job. Like, your job is, yes, to kickstart it, but then you need a plant that's able to do it. And if the plant doesn't have adequate boron, that whole system's not working. And you think about how many tens of thousands of pounds of sugars could be being pumped through those root systems. You putting out like a cup of sugar, or whatever you're putting out onto your land, is not the same as what a plant's actually able to do if you support it. So for me, boron is one of the critical elements that we have a look at, and we look in plant tissue tests, not the soil. Have a look in the plant and see, is that plant got optimal boron? Is it able to be pumping sugars out through the roots? So we're putting small amounts of trace elements in. It might be like 300 grams, whatever that is an ounce. I don't get American ounces or something like that. Small amounts of minerals. And then we're addressing what we call sleepy soils. So we did some microbial inoculants on the actual seed, and then look at hygiene management. How do we help break down some of those and digest some of the stubble that's sitting there? So when I arrived, there's stubble in those soils that's probably 4 years old. You see in this photograph, that stubble is all last season. This is canola coming up, and the other thing I want you to note, if you can kind of see in this picture, is how clean that field is. So yes, they use a pre-emergent herbicide, but when those plants come up, they have the biology around them on those on the seed dressing, and we're stimulating biology, and we end up with cleaner seed beds than if you were to just try and come into the soil and transition it. So the other part of addressing the limiting factor was looking at management, so some of the crop choices, looking at increasing diversity and trying to get cattle over. But when you're on big operations like this, so this is 30,000 acres, it's just not possible to get cows over every acre. And I mean, maybe it is possible, maybe someone can do it, but these guys currently aren't getting cattle over every piece of land. We also look at how do we remove or replace things that we know are really harmful. So those neonicotinoids, seed dressings, they just got to go. You don't want to be putting a seed down that has pesticides and fungicides on it, because it disrupts not only what's happening in the soil biology and everything else, but actually disrupts plant physiology. And this has been some of the cool stuff that's coming out now too, is the impact that those seed treatments have on plant health is just massive. So it creates a plant then becomes more vulnerable to pests and diseases. So it's a perfect sales model. Use my seed treatment or my pesticide, and you're going to need to buy more pesticides off me. So removing or replacing some of those harmful things. So we pull out the soluble phosphate, for instance, pull out those things that we know are detrimental to biology. So this is what the soil looks like when we arrived. So those roots really aren't penetrating below that 2-inch zone, and you see they just grow sideways. So that means, and this is actually typical for most production operations around the world, is they're really just farming in that top 1 or 2 inches. So this is why we're droughty. Your root systems can't get down, so suddenly you aren't able to access water and you're not accessing nutrients. So Kay Baxter might have talked about the loss of nutrient density in foods. So we're seeing a decline in what you're eating, and we certainly see an improvement as we start to access that whole profile. We see a massive lift in trace elements and minerals into the plant, and then that reflects in terms of animal health. So we measure that in meat in terms of what is it that we're actually eating, and that is influenced by how deep are these root systems getting down. So I don't know if this video will work. Riley, is it working?
Not working on my end. Probably have to forego the videos, and we can send people a link about the videos you refer to in the replay.
So what's interesting about that video is that they put in a cover crop, but to dig up this, so this is sunflower root here on the left. I mean, look how cool that is. Those things really do penetrate through soil, but we had to use basically a pickaxe to dig up the soil because it's so tight. It's just so impenetrable to work a way to get down there. So these cover crops are certainly a powerful tool. They are going to open stuff up, and their roots will, they're going to die down and add organic matter. But it's hard for the biology to be moving through a system like this. Like everything's just pushing, and it's hard work. And so the photo on the right, they only got a third of an inch. They got 3 centimeters, so that's just over an inch of rainfall in 2017. So that's not good. that's not good for anybody if you're getting rainfall like that. So what they had was the cover crops actually failed as well. And so you just got to watch with cover crops. Are you getting it in with adequate moisture? It's not a silver bullet, and it's what I'm starting to see as people are treating some of these tools like silver bullets. It's a whole system approach, right? What is limiting what we're dealing with? Okay, so in their first year, this is basically their program. So down the slots, down with the seed, we put a humate prill. So humate is your concentrated carbon, and we've put it. So a prill is just like a fertilizer, kind of a little pellet. We combined that with gypsum. So it was about 35 pounds of gypsum and 30 pounds of humate with trace elements and a bit of sodium were in that prill. So that's going down below the seed. And then what it does is as a slow release, the gypsum reacts with that high magnesium, makes it soluble, washes that magnesium out, and replaces it with calcium. So we start to effectively what we call flocculate. So we start to open up that soil. Now that soil can breathe. Now microbiology can get through. Now those roots are penetrating, all right? And we put a seed treatment on as well. That particular seed treatment had a phosphate solubilizing bacteria, azotobacter, which is your nitrogens. We did use a mycorrhizae, although I'm not confident that the mycorrhizae was actually that effective, but we put a mycorrhizae on the seed too. As a foliar, we put 2.4 litres of an NPK, so very small amounts, like we're talking, so we're talking litres here, so sorry guys, it's about half a gallon, just over half a gallon. So very, very small amounts of NPK, and in that with a fulvic acid. So fulvic is another component of your humate, so it's a soft brown coal, very, very, very fine surface area. So we're talking about a chelating carbon. So basically that carbon gets hold of your trace elements and your NPK and then enables that to slow release as the plant needs it. So we're gathering up the inefficiencies of, you know, people just putting NPK on without considering you have to feed the biology. It costs them $120 less an acre. We reduced pesticides by 95%, and it was just that one, they couldn't get seed that didn't have a seed dressing on it. And we reduced the herbicides by 30%. So this is just year one. So these are what I call transitional programs, or some people will call the methadone program. How do we get off your high input system profitably with, as far as I'm concerned, no risk? So this is a no risk program. We can put this into anybody, anywhere in terms of cropping, to be able to just start transitioning them off. So you're probably thinking, where's the nitrogen? So they were putting a lot of nitrogen in, so 110 pounds of actual N, so that's like 250 pounds of urea. We dropped that immediately down to 30. This year we did 25, and next year we look to be maybe putting down 2 units. But we will be monitoring it to make sure that nitrogen is working. But what I want you to just notice in that photograph is just the width of that wheat, just how strong that little plant is, how it's got, how soil's all stuck to those roots, and how those roots actually are going down. So within 3 weeks, those roots had penetrated through that 2-inch hard pan. I've got some photographs of it. And so we're getting over what their main limiting factor was, which is those roots couldn't penetrate, and no matter what they were doing, the roots wouldn't penetrate. So that's canola on the left, barley. So then this is one of the things we're looking at. What's happening in that plant? What is that plant experiencing? Is it getting all the nutrients that it needs, and are these nutrients in a balanced ratio? So I don't need to get into the different figures. The colors are probably helpful. So you see things like sulfur, magnesium, and calcium were in excess, boron and copper were deficient. So within 1 year, we bring that all up into balance. So now that plant is actually, it's less stressed. It's going to start pumping more carbon. It's going to pump down more complete proteins. We're not attracting insect pests. We're not going to have disease issues. Those plants are now taking care of themselves because they have all the nutrients that they need in order to do that. And biology is what's helping them do that. So it's a cycle. Well, it's not a cycle. It's working backwards and forwards. Feed the biology. Plants feeding the biology feeding the plant. No one's independent. That's their lovely peas, looking very nice. So anytime that we're using chemicals or nitrogen, we're always putting a carbon in, always, always, always. We never do any additions or any actions, so that might even be ripping or drilling or anything that we're doing to disturb soil, we always put a carbon in. And fulvic and humic, I find, are just, they're easy to use, they're not very expensive. And so we're adding 1 part fulvic acid to 4 parts of their herbicide and reducing the herbicide use by 30%. And I know some people in this group probably are like, you know, we don't want to see any herbicides at all. That's a good goal, all right? So that's where we're heading towards, but on 30,000 acres, we're not going to be crimping an entire cover crop, for instance, or and maybe we will one day, but when we're working in transition, this is a transition tool. Okay, so yes, I'd like to see the herbicides out, but what's interesting is people have been doing this for a while. You can test your crops and they have nil chemical residues in their end product. They come out cleaner than what some of the organic products are coming out now in terms of glyphosate, right? It's not in the end product. Okay, so in year 2, what we started doing, oh, that played, is we put in an extract. I wonder if I can, oh no, I can't mute that because I'll mute myself. So we basically put in a worm extract, went down with the seed, all right? So within year 1 and 2, we got those roots down through the hard pan. We're seeing healthy crops. We saw equivalent yields. We're now seeing resilience because those root systems are getting down and it's incredibly profitable. I mean, I could tell you how much money they're making. It's ridiculous. Through looking at how do we address what our limiting factors are. So the interesting thing for me is, so we're putting down half a gallon of worm extracts in here, and I was going to show you another case study, but I think we probably don't have time, is that we're putting just small amounts in, and how is it that we can have small amounts of like a worm extract or a compost tea or a compost extract? How is it that they're so effective? And we've known this, I mean, yeah, there's people working on this, but there hasn't really been the science to come in behind and explain until we started to look at what some of these signaling molecules are. So for instance, there's tens of thousands of different types of signal molecules in soil. So they're proteins and hormones and enzymes, elicitors, yeah, different types of your peptides. So these are called quorum sensing molecules, some of them. So what they are basically molecules that switch on activity on and off. So we've known about quorum sensing for a long time. Since in the 60s, they were looking at it in terms of inflorescence in deep sea squid and things, right? So the bacteria switch on and switch off. So it's a signal that enables biology to turn on and off. So what we're discovering is this is what's driving plants and biological communities is this type of signaling process. So they were looking at it in terms of human health and how to kill it, right? Because if you're going to be looking at things in science, it's going to be, you know, something you want to kill. And so at any one time, you could have strep throat, for instance. You could have the organisms in your throat, but you're not virulent. Well, they get to a certain point and they switch on a signal and turn themselves virulent. Now you're sick. So the scientists were looking at, can we find the chemical signal to turn it off? So for instance, they found a chemical signal to turn off cholera. You can drink this chemical concoction and the cholera that's in your body goes, oh, there's lots of other cholera here. I don't need to be here anymore, and it'll actually leave the host. I don't know how close that is to being commercially available, but this is where some of the research is going. What are these different chemical signals? And what we're finding is in this root zone, as you start to get more chemical signaling, biology, you're able to express different gene profiles or different, what they call housekeeping genes, that they can't do by themselves. It takes a community for them to be able to establish full genomic expression. It takes multiple different species interconnected in these communities. So it's how stable are these communities, how much biodiversity do you have, and how much biomass do you have for these signals to start to happen. And this is the basis for nutrition, and this is the basis for resilience. So I could keep going on, Rawley, but I think maybe we could have a conversation, or you could ask me some questions because you've probably got
Sounds good. Let's do it.
Yeah.
So yeah, there's a few folks who asked questions at the beginning. Before we go on, yeah, we're gonna do a little question break for folks. See if Andrew Langford's still here. Now, Andrew's is this the same Andrew who runs the Gaia Institute. So he has this really cool thing called the Canvia solution that the Gaia Institute has been working on. Where they have like a compost reactor that outputs really high fungal compost, and we got to
Yeah.
get him on a webinar because that's pretty awesome.
Wow.
But he asked about an origin of microorganisms question. Is it better to buy commercially produced microbe to inoculate soils or cultivate these using local parent stock?
Yeah, local parent stock for sure. But what I'm finding is when we're in transition for some of these guys early on, they don't have access, and it's really hard to get hold of really good quality high fungal compost. And then within year 2 or 3, we're starting to make our own extract. So my goal would be, yes, let's get the best quality inoculant that you, not inoculant, but like compost or extract source, make it yourself using indigenous microorganisms. So say for instance, like you're dealing with a glomus species, like a mycorrhizae, although it might be the same species all around the world, its ability to have different genomic expression based on its environment that gives it an opportunity in its environment. And I just feel like we really don't know enough in terms of introducing microbes that maybe they've come from India, because India is certainly making a lot of these inoculants. But yeah, I'm finding in our transitional phase we are buying some of these microbes, like there's entomopathogenic fungi. So these are fungi that live inside plant roots that kill insects. We are using some of those because it really helps speed up our program if the microbes are being eaten away. Sorry, if the roots are being eaten by microbes. So yeah, it's always a case-by-case basis. I would prefer to make my own, and my own compost and my own vermicast. And if we can't, yes, there are packaged products. That didn't really answer your question.
I think he did a good amount answering that. His follow-up question was that he was like, will exotic source biology conflict with the local strains if they're used together?
But they can. And so if you looked at like the early soybean, when they first started developing inoculants for soybeans and they put them out, that was the inoculant they used, the type of rhizobia. And 10 years later, they developed a better rhizobia that would make more quality, and it won't outcompete with the original inoculant that they put out there, which creates lower yields. So by not kind of really understanding what the species was that was put out, they now have reduced yields in the US because of this original inoculant. And that is possibly something we're risking by some of these mycorrhizal inoculants that people are using. Yeah.
Okay, cool. So here's a New Zealand-based question. This is from Kate Bradloff. She's saying, I'm on the top of the South Island of New Zealand, and I'm surrounded by quickly increasing numbers of hop farms. Do you have any experience with hops? And do you have any advice as conventional hop farmers here, not really focusing on soil at this stage?
No, and it is, yeah, it is scary. And I guess with hops, there is actually quite a good opportunity to put in some kind of cover crop underneath the hops, and a lot of people aren't, so they're just spraying it out and they're all concerned about competition. And so yeah, I mean, a lot of those hops are basically, it's almost like hydroponic farming if you look at how almonds are grown, for instance. And so you're basically just propping up the system with fungicides and pesticides. And yeah, like it is with every industry, I don't think hops are unique with that. We need to be really looking at the whole system. I had a farmer recently say to me how he felt like modern agriculture was a broken down truck, and we're just using different trucks and different more mechanical solutions to try and pull that broken down truck. And that's what we'll be seeing with the hops. So yeah, there's no quick answer to that one. Yes, they need to be looking at soil health.
All right, fair enough. So here's a question from Joe saying, do you have any recommendations on which lab to use, preferably in BC, for Canada, for sap or plant leaf analysis?
Yeah, so for plant leaf analysis, you could just use A&L labs, or most of the labs in in Canada can do just normal leaf tissue analysis. It's quite a common analysis. So when you do it, you want to get a complete test. So you want your mineral profile, and then if you are feeding animals, you do want that complete profile in terms of NDF and crude protein. And one thing I really recommend people do is get a plant tissue test of the plant that they're trying to grow, and then if you've got a lot of weed pressures in those fields, to get a plant tissue test of your weeds. And I'm sure there's a lot of labs that have just been really pleased when they get packages coming from us full of thistles. But you want to look at line those two tissue tests up and have a look at where is that weed significantly higher in an element. And generally it's because it's a dynamic accumulator. It's bringing nutrients from deeper down. And so it will, it can be a very powerful indicator for what's going on in terms of soil and plant health. So yeah, do a tissue test, then do a tissue test on your leaves, and do a tissue test on a weed. It's really, really cool stuff.
All right, cool. So here's a question from Joe asking, is there a difference in performance between using dried vermicast versus fresh in treating hydrophobic soils?
Yeah, the fresh is going to be better, but you're gonna struggle to get the fresh stuff down, like it won't go down the drill as easily. You'd need to put it into solution, so then we'd apply a slurry. So if you've got the ability to spray slurries, then I would use vermicast in its wet state. However, what we're looking at is these quorum sensing molecules, so it doesn't matter if it's dry or wet. It doesn't matter even if, like, you know, we talk about we can't put compost teas with trace elements, for instance, because it'll kill them. You know, I'd like to have living biology out there, would be great, but if we kill the biology in the process, we still have these quorum signal molecules going out. So the answer is yes, fresh would be better, and but if we have to use dry, then yeah, we can use dry.
Okay, now here's a big question. This is from Marvin. He's asking about, so this is why if Elaine was on, she'd probably like raise her hand and ask this. This is Marvin's saying, Dr. Elaine Ingham says all the minerals are present in the soil. You just have to make them available, adding the right biology. Marvin's saying, are you saying that we do need to add some certain minerals to kickstart the system?
Yeah, that is what I'm saying. That in some cases you're going to need a catalyst, the mineral catalyst. So say for instance molybdenum and cobalt, trace elements that might not be readily available when you start. They're both essential catalysts in nitrogen fixation. So the free-living nitrogen fixers, just like the rhizobia in the roots, need those trace elements in order to form the enzyme that does the nitrogen fixation. Now, if you're critically low in those two elements, nitrogen fixation is not happening. Even if you put nitrogen fixers out there, they can't do it without those two trace elements. And so if you don't address those trace elements just to kickstart, then the biology can't do what they need to do. And so yes, I do believe that biology will provide all that we need eventually. All right, so how quickly does that process happen? It's not an overnight process. So in some operation, microbes are the limiting factor and all you need is biology. And in some operations, like if you look at New Zealand, for instance, it has a huge amount of biology. It's got a lot of rainfall. It's got a lot of organic matter. Biology isn't necessarily the limiting factor in New Zealand. And if all you do is put biology out there, or all you do is put biological foods, you can crash your system. So I've seen people, multiple people now, crash entire systems by just trying to feed biology. You've got to really look at what is it that's putting that drag on. And if you've got major compaction issues, where is the biology that you're putting on going to go? So it's is biology all you ever need? Yes. No, maybe, perhaps, you know, over what length of time? So I'm really interested in how do we kind of transition quickly, and microbes might not be the only answer.
Cool. No, that's great. So we've had Elaine a few times, and she's pretty adamant that this can happen, you know, chemical-free transition in a single growing season. And there's been some case studies where she's gone, this can happen in a single growing season, but you know, you're showing like sometimes it's not just a single growing season.
That's right, and that's because on that operation, microbes were probably their limiting factor, but
Cool.
not all are. And so that's the benefit of kind of working in lots of ecotypes, is we can see in some environments that is the case, and in some it's not. So you've got to really look at your own specific environment.
Cool. So Svetch is asking, what about labs in Europe for testing soil and plant tissue analysis?
I don't know. I'm not working in Europe.
Okay. Svetch, there are a few like soil food web labs in Europe.
Mm-hmm.
Like, if you email me, Raleigh at Sustainable Design Masterclass, I'll send you a link, because there is soil food web analysis in Australia, Europe, and North America. So
In New Zealand.
Yeah, New Zealand.
Mm-hmm.
So Robert Porter he's saying, I see a lot of variation in the quality and effectiveness of commercial inoculants, especially AMF inoculants in compost teas. Can you recommend reliable directions from certified manufacturers?
Yeah, and I think this is unfortunately one of these buyer beware things, because there's so many products coming out into the market. I mean, even New Zealand has something like 160 different companies now supplying different microbial things. And a lab here in New Zealand tested the mycorrhizae, and out of 7 of the most common commercially available mycorrhizae, none of them were viable. You've got to ask sometimes how those labs are doing those viability tests. So there's different types of viability tests, and it will depend if it's spores or actually pieces of mycelia. And so they have different lengths of time that they need to incubate. So you need to really don't just take a viability test. You need to know the method that a laboratory used. So it's very much a buyer beware thing in terms of you need to know what questions to ask. I always look to what have been the success of producers in the field. So often if I'm looking at using a product, I'll talk to a grower or a producer that's been using that product for some time and has actually been doing the measurement. So I want to see, if you're using a mycorrhizae, can we test that in the field, and is it actually showing up that it's improving colonization? So side by side trials where people are using mycorrhizae and not using mycorrhizae. And so for me, it's actually ruled out quite a few big name commercial products because they're not effective in the field. Yeah, so I wish there was a simple answer to this, and I think it's probably where the industry is going to have to go in terms of starting to regulate. Are you buying talcum powder full of dead stuff, or is this product actually viable and working? So we want viability tests from those companies. They need to produce a viability test. Yeah.
Fair enough.
Mm-hmm.
So Tatiana's asking, I'm working on increasing organic matter. What other types of methods would you use for improving very sandy soil in order to retain nutrients and water?
Yeah, so you want to be speeding up this process, I guess, and that's where actually, I mean, it depends on your scale. If you can actually put solid amounts of compost on and solid humate applications, and then getting cover crops in to really build biomass, yeah, that'd probably be really good kickstarters. And then it's looking at your plant's bricks, or how well your plant's photosynthesizing, as to how much sugar is that plant pumping out through the root zone, because it can be significantly more than any compost application you're putting on. Yeah, I always think of compost as kind of like, I'm here for a good time, not a long time honey cycle, you know, so it's kickstarting that process and supporting plant health so the plant can start pumping sugars deeper down, because what builds carbon in soil is microbiology. It's not the addition of organic materials like compost. Humate itself is quite interesting. If you think about it, it's a prehistoric woody vegetation that's just been packed down over a millennia. It's full of these quorum sensing signals, I'm pretty sure. We're waiting for some of the research on that, but so it's part of what's signaling fungi and biology to really get going, and then they're going to be consuming those plant sugars, and then they start building organic matter. But I think absolutely getting cover crops and making sure you've got a living plant root all year round will build your carbon.
Nice. Okay, so Jay's question is, how far is too far away to get compost? Same state, but purchased from dry and colder areas in the north to more steady, hotter areas further from the south of the state.
Oh, wow. I don't know. I guess you're going to be looking at what's the cost. Are we talking about indigenous microorganisms, are we?
Yeah, Jay, maybe rephrase that a bit.
Yeah.
Okay, how far away is too far away to get compost? So one area is too cold and the other is too hot. Like,
Okay, well, too cold, we can still build good compost. Too hot, yeah, you're going to have different challenges. But yeah, I mean, I'll travel interstate to get good fungal compost. I mean, we've been getting some out of Oregon and and taking it into Montana. And if I was bringing compost in from other areas, I would probably mix it with some of my indigenous, like either duff, if you can find some stuff from underneath the forest, or finding some indigenous microorganisms to incorporate it, which comes into your Korean natural farming too. So making some of these beneficial indigenous microorganism brews that you can get out of the forest, and then putting that into your compost, or from where you've got really healthy prairie lands, using that as an inoculant. But yeah, I will travel quite far for good compost.
awesome. Yeah, he said he's looking at compost being made by Elaine students in the Taos area. He's looking at Las Cruces in the hotter south. He says not many qualified people making it down there.
No, and it is unfortunately really frustrating, but I think that's where I'd be just make your own using David Johnson's biodigester system. It's really, really easy to do, and everyone that I've seen doing it has made like a 10 out of 10 compost. Like, it's crazy. People that have never made compost before using his Johnson Sioux biodigester are making really good fungal, high nematode protozoa numbers. I guess when you're wanting to make an extract, you really need to have basic ability to look in a microscope and have a look and see if is that good quality, and that becomes another stumbling block. You need someone to hold your hand, or yeah, hopefully you've got the skills so you can assess is that good quality compost, because there's a lot of garbage on the market.
Yeah, damn straight. That was one, like, we were doing a microscope workshop 2 weeks ago with Elaine, and they're looking at, there's a local like worm casting compost, and they left it just baking in the sun. They're looking at it, and they was like, well, there's no biology here.
Wow.
At
Nothing.
all. What a surprise.
So yeah, we were looking at compost that some of the best compost I've seen has been from the mushroom places, so mushroom compost that hasn't been sterilized. But what they do in some places is they treat it with methyl bromide. So you like go, oh, I'll get some mushroom compost, it's good, and then it's like, oh, it's treated. So yeah.
So here's a question from Ken concerning dairies. He's saying, have you worked with dairies? We're located in the most important dairy area in Mexico, actually a desert. We have very bad soils, high in pH, salts above 6 ds per meter. We have even more than 10, low organic matter, etc. Do you think we can recuperate these soils? Most of the crops are all irrigated by flood.
Yes. Yeah, I mean, it's making sure your flood irrigation is managed well is always a challenge. But yeah, I'm working on quite a few operations where we are rehabilitating saline soils. So if you're not sure what the answer is with a lot of this stuff, the answer is carbon. So if we've got a saline issue, more carbon. If we've got not enough of something, probably more carbon is the answer. So we're using worm extracts to actually shrink saline conditions, so putting on higher amounts, so maybe 10 gallons of, I keep talking in gallons, sorry New Zealanders, so 100 litres a hectare, 10 gallons of like a high fungal compost in a worm extract to just support that rhizosheath, right? So that plant can actually get itself going and buffer those saline conditions. But really interested to hear about dairy operations in Mexico. I can't even imagine. But yeah, I guess if you get irrigated, you're growing some grass. But these things are possible wherever we are. It just comes back to putting quality back into the system and getting that buffer. And if you're irrigated, you've got a huge advantage in terms of being able to build carbon if you're irrigating well.
Nice. Okay. Kara's asking, what's the name of the biodigester guy? It's Dr. David Johnson out of New Mexico.
Dr.
Right?
David Johnson. And if you Google just on YouTube, it's Johnson-Sioux SU biodigester.
Johnson Sioux biodigester. Yeah, that's pretty cool. We might Might have him on like a
Oh, yeah, you totally
Elaine
should.
David Johnson like co-webinar, because she was saying that he was increasing the carbon uptake to 12 tons of carbon per acre.
Yeah.
Like that was kind of the high end, and that's pretty huge. Like not even forests are getting that.
No, well, I mean, yeah.
Not meant like there's some forests that aren't getting that.
Yeah, totally.
Yeah.
No, what he's doing is just phenomenal. And I think he's showing as well that it's, we have a respiration cycle. So, carbon's always in flux, and he's dropped that loss of carbon to the atmosphere down to 20% of what it is without all of that high fungi. So
Wow.
he's doing some really exciting stuff. And that's what we need is just those guys supporting. How is it that we're seeing this in the field, and how some people are achieving the results that they are? And then, get people like Dr. Johnson really looking into it and finding the mechanisms is exciting, because I think it's giving us more of a foundation in discussing these things with the conventional guys.
That's such a huge potential, because once that, if any country has a carbon price too, then you can show it's like, okay, we're increasing the sequestration rates to 12 tons an acre. Imagine these people with large farms. That's a whole new source for them to reason to do this too.
Yeah, and there's a really cool project that's just started in Montana where they're paying ranchers up front for carbon, and paying them for behavior change, so putting in fences and putting in more water for adaptive managed grazing, and they're paying them, I think that they're guaranteeing them $3 a ton per acre. So, I mean, if these guys can do 15 tons an acre a year, and then they pay them in 5 years' time, and they're on big areas, we're talking about a lot of money, and that's all in the carbon market stuff. So I'm excited to see that stuff, and we're already using some of these biostimulants out there on rangeland. So I think we're going to be seeing some really good secondary carbon measurements coming in.
Nice,
cool.
That's awesome.
Yeah.
I'm curious, like a lot of the people, most of our webinars we've got the biggest response for two things: people interested in restoring the soil and has that improves yields, but also how we can reverse drought by restoring soils. So how, in what kind of areas have you seen horrible drought conditions reversed by repairing the soil? Like, and how does that dealt with extremes of weather, where it's like extreme drought and extreme flooding?
Yeah, well, I mean, this has been the fun stuff, really, is that we are working in a lot of extreme environments. So guys that are in rainfall zones, that are around that 7 to 9 inch rainfall, so they really are those semi-arid environments, and just seeing this base density of green pasture now starting to come through has just been mind-boggling. When you ride, I like to ride, so you're riding around and it's just dust with the neighbors, and then see what they're achieving is just amazing. But yeah, I mean, I think you still need things like germination rains. So if you don't get those early rains in some of these big cropping areas, we're still in trouble. You still need a little bit of rain if you're putting down annual crops, but for the perennial guys, it's really just getting that root system down and building resilience. And it is that buffer. Carbon is the buffer against these temperature fluctuations. And they did a neat study in New Zealand on the East Coast, looking at holistically managed operations that had tall grass cover that still had a green pick in it after 6 months of drought. I know people are going to go, New Zealand doesn't have droughts, but dry. And just looking at, actually we can keep a good base of grass if you're managing well for drought. So yeah, I think it's providing a catalyst. That we're seeing some really interesting conversations coming out of Victoria, New South Wales, and Australia right now, because they have to start looking at regenerative. We have to start building carbon and building that foundation. Otherwise, you're not going to be farming there in the future. It's unaffordable. And yeah, so yeah, there's good case studies wherever we're going right now. You can go and see operations that are showing that drought conditions can be mitigated.
For those in Australia and New Zealand, where do you reckon some of the top places people can visit to see soil restoration case studies in action?
Well, I guess a lot of these guys are getting a lot of attention, so sometimes I feel like, do we want to keep throwing the same names around? Because, I mean, a couple of my guys just changed their phone numbers because they're getting over like the amount of attention that they've started receiving. But yeah, I think there's some really good case studies in Western Australia for sure. And I mean, I always talk about Di and Ian Haggerty because I think they're doing an extraordinary job, and their stuff has been backed up by some pretty good CSIRO research in terms of water holding capacity and carbon, and they're using worm extracts, and certainly seeing some of the stuff that Cole Size is doing in Australia. I really like Dean Martin in New Zealand. He's doing cover crops and has been doing holistic management for, I think it must be close to 20 years in New Zealand, and just really showing what's possible in areas that do get a big dry. But yeah, there's a big list of people I can mention that, yeah, I think they're doing a neat job. But, yeah.
Well,
but
awesome.
I don't think New Zealand or Australia are any further ahead than anywhere else. I just think that both of them have smaller populations, so it's easier to get in contact and like have a sense of community. You know, there's so much stuff happening in the US and in Canada. I mean, Canada, what's happening with cover crops is just extraordinary. Like, it's just booming, and you've got extension groups that are really supporting information, which doesn't happen everywhere. You know, we don't necessarily have those good extension, and there's certainly not in New Zealand. Like, there's no extension services except for, say, the conventional beef and lamb or the dairy NZ captured kind of information. Whereas Australia, their land care groups and their no-till groups, and even the government are getting in behind things, so it's easier to get information and go and see different farms. But yeah, I don't think any place is behind any other place. It's just the case studies that are coming forward.
Yeah.
Mm.
And New Zealand, oh, there's places I got to, like Kate Baxter's place, the Coang Institute.
Yeah.
Oh, it's just such a lovely place because they're bio-intensive gardening everything, and though I don't think they're looking at a microscope, it's so, like, soil is so deep there, and
yeah,
they're testing to make sure the bricks are getting up there on the plants.
so
Okay.
great.
Awesome.
Okay.
Well, I think we're about at near an hour and a half here. We could get going unless you wanted to share something else real quick. Do you think, is that pretty much everything? Was that everything you wanted to share?
Oh, no. I want to just show one thing if I can.
Sure,
Because,
go for it. Here, I'll have you share your screen. I'll change the presenter to you.
Because this is one of the things that just blew my mind, and it's only just a couple of it's just a couple of slides. But can you see that?
Yeah, I can see it.
All right, so this is Di and Ian Haggerty in Western Australia. So this is second year into taking on very, very degraded soil. So these soils, they took these properties, they were just filling them full of herbicides. They really didn't have a way of effectively managing the weed burden that was on this property. So huge amounts of long-term residual chemicals were being applied in here. There were areas that have saline problems, some pretty gnarly weeds. So what they do is they're putting down worm and compost extracts down the drill. They're not doing anything else. They do a pre-emergent herbicide, but at probably 50% of the rate of normal herbicides. So what was interesting is in the first year, what started to come up after they harvested their crops were what we call early successional grasses. So in Australia, these are your button grass, your kerosene grass, your windmill grasses. So low palatability, not stuff that people really want to see, but it was ground cover, right? So these guys are looking at how do you have something growing over summer? So they grow crops in winter here in Western Australia, and then in summer you want ground cover. Well, what was super interesting is in year 2, what you're seeing growing here is called Paspaldidium, and it is a native C4 grass. It hasn't been seen in this area for 60 years. It looks like someone's sowing it. Like, it's just mind-boggling. So this seed is germinated based on the signal that it's receiving from the worm extract naturally. So now they don't have to plant a cover crop, they actually have a cover naturally coming in and protecting that soil. So if you think about herbicide resistance, there's now over 300 weed species that are resistant, and it's the same with antibiotics and pesticides. All right, we're facing a perfect storm of pesticide and herbicide resistance. Right, it's just the arms race. It's just ridiculous. So this is the global increase in unique resistant cases to herbicides. So it's just on an exponential rise. What these guys found, and I just have to explain this to you, this is the herbicide resistance in the ryegrass seed. So the ones at the top, the green and the blue, this is what the seed was before they started on their program. So 2 years ago, you had glyphosate and trifurulin resistant weed seed. The purple and the pink is the weed seed that is now in their soil. So they are now able to kill what was previously herbicide resistant. They're killing at half the rate of herbicide for glyphosate and trifurulin, not that they're using that. But this herbicide resistance issue that we're seeing is a biological issue, and we can address it through these quorum signals, through soil health. We can actually deal with herbicide resistance, because I don't know if most people in the cities probably kind of aren't aware that this is happening, but herbicides resistance is massive. And so if you're working for Bayer, then you're going to be looking at, okay, we need dicamba now, we need to mix 2,4-D with your glyphosate, and now we need blah 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 blah. They're having to put all these chemicals in together to deal with it, whereas these guys are doing it in a 2-year period at half the rate of chemical. They're changing the signal around the seed, and they no longer have those weeds germinating. So that's just something I wanted to share that I'm really, really excited about.
Nice. Well, yeah, that's a good way to end it on. Unfortunately, we got to get going because we're sharing the webinar with the Ecological Action Team community. They've got another webinar coming up in 4 minutes. But Nicole, that was fantastic. Thank you for sharing everything.
Thanks for having me on.
You got it anytime. And for folks who are interested, look in the chat box below. She's got a site, integritysoils.co.New Zealand, which you can get in touch with her for if you need a consultation on how to biologically transform your land.
We've got workshops in New Zealand and Australia and North America in the next 6 months. Awesome. So we get around. So right, thanks, Rory. Yeah.
All right. Well, thanks, everybody. Unfortunately, we got to get whoosh, cut it off abruptly. But thanks for tuning in. We're having Byron Joel and another awesome Australians coming on to share kind of the ecological history of Australia and what we can learn from it next week. So stick around for that and have an awesome weekend. Peace, everybody.
Peace.